Good afternoon students, molecular genetic. In live class we are discuss about what is transcription and functional unit of DNA and transcriptional unit of gene. So this and all we already seen. So we go for the new topic. After that what do you mean by transcription and DNA, everything you have studied. So we will see in the what are the process involved in this, that is strand, and everything we can study in this structure. Okay, so the transcriptional unit and the gene. Okay, this is the functional unit. So transcriptional unit in DNA is defined by three region. Okay, how many region? Three region as a promoter and structural gene and the terminate. Okay, the promoter of the located towards 5 end only. The DNA sequence that provide a binding site of the RNA polymerase. The presence of the promoter in a transcription unit define the template and coding strands. So the termination region located towards the 3 end of the coding strand contain the DNA sequence of the transcription. So see, this is the transcription unit. So in the template strand, this is the coding strand. So it's a terminator. Terminator means end. So 3 from to the template strand. This is a center is structural gene is there. So the orange color is the promoter. This is the terminator. So in the promoter will be form a structural genes, and that terminator will cut the end. Okay, so then it will be stopped at the transcriptional unit. What is that? It is a cut the transcriptional unit. This is the main point for this. Next, the DNA template, you know that the thymine and guanine, adenine, cytosine and adenine is present in that. So it is a minus 35. This is the 35 region is that. After that only one set of that. This is the print now box and strand of the RNA. This is the start of the RNA. Okay, so RNA here only started. So DNA template will be formed. So see that what is the difference between the RNA template and that. This is the Hogness box. This is sometimes present. So it is a differently way in the eukaryotic promoters only. This is the first one strand is the prokaryotic, second one is the eukaryotic cells. So E is the promoter side. So what is the difference between him? See, check it that it is a 75 region is one meant CGN. So that is the cytosine, guanine, niacin is present, cytosine is there, adenine, thymine, cytosine here. So this is the 75 region in the eukaryotic promoter cells. So eukaryotic cells only. So, okay, and they see next is the some Tata box is there. That is a Tata box is that what is a thymine, adenine. The two only is present that. Okay, so this is the sometimes guanine also is a present in that. So this is why it is a promoter cells is a form like that, means define the template and coding strands. The termination region located towards the 3 end. What is that? The coding strand contain the DNA sequence that causes the RNA polymerase to stop the transcripting. So RNA polymerase help to stop the transcripting. So in eukaryotic the promoter has a adenine thymine rich region. So that only adenine thymine is present more than, means it is a Tata box, otherwise Hogness box and golden bag box. This is a scientist name. Then only they give that he only noted this Tata box. Then only her name they are giving that. Okay, so prokaryotic region only it is present, and because it's present in the prokaryotic means it is called as a print now box. Beside the promoter, eukaryotes also require the enhancer. The two strands of the DNA in the structure genes of transcription unit have opposite polarity. DNA dependent RNA polymerase catalyze means to fasten their reaction, the polymerization in only one direction only. So template strand and now originality strand is called as the template strand. Okay, polarity 3,5 act as a template, so it is a template strand, and polarity 5.3 has a sequence end, so it is displaced during the transcription. Because why means instead of thymine here what is the present? I already told thymine is only DNA, uracil is RNA. So this strands only calling a coding strand. See, this is the transcript at last minute what I told that only that promoter we can cut it and show in the base sequence template and coding DNA strand and the base sequence of the mRNA transcript. So mRNA means what is the, instead of what is it present? Thymine, uracil here present. This is the difference between. Okay, this is the 3' to 5'. We can cut it the once a set of DNA region we can cut it and show it into the picture. Okay, next the structure gene may be the monocystonic. From where? Eukaryotic cells, and polycystonic. Many it is the prokaryotics. Okay, in eukaryotics mRNA carries only single gene because it is a mono. Mono means single. Here mono means many other single proteins are present. So the prokaryotic clusters are latent gene known as the operon, and next to each other and the chromosome and the transcriptor together to give a single mRNA, and hence are the polycystonic. What is that? Polycystonic. See, this is the transcript template. The red color is we call as the protein displacement. So normally we can straighten that to, because the DNA is a hexohelical, we can straighten that and show it that here. Okay, adenine is this is the translation strand, and protein 1, protein 2, protein 3. Okay, there is mentioned in the different colors of the proteins. So it is everything is a translation start statement. So this is the RNA. Why miss it is RNA? Because here uracil is present. So it is a RNA, eukaryotic RNA. So that is easily you can understand that. DNA means what is that? Thymine present. What is that present? So uracil only present in the RNA. Okay, thymine present in the DNA. Before starting the transcription, RNA polymerase bind to the promoter, recognize sequence in front of the gene. Bacterial RNA polymer consists of two major components. The core enzyme for RNA synthesis, it is a sigma subunit responsible for the recognition of the promoter. So it is a maybe vary in the promoter sequence only. So see this general stress, heat shocks and all, the promoters and all with the prokaryotic RNA polymerase. So it is will be changed in their direction. Okay, so see next RNA polymerase open up the DNA to form the transcription bubble. The core enzyme moves ahead manufacture RNA, leaving the sigma subunit behind at the promoter. So the end of the gene is marked by a terminated sequence that forms a hairpin structure in the RNA. The subclass of the terminator recurs the record protein known as the, so it is the property in the function. Okay, so next process of the transcription. See, this is the process of the transcription. So here RHO dependent termination. Here the termination end is there. So rewinding. So it is a transcription bubble. We form then so rewinding this structure. This is the 3 frame. This is a 5 frame. So in that way only we are show in that. So this is the unwinding sequence. So this is the normal. You can see the double helix DNA. So we can rewind again. We are unwinding this structure. So because of the RHO dependent termination, cut it that we can cut that termination. The polymerase transcription away from the promoter. RHO factor bind the RNA, follow the polymerase. So the RHO factor helpful to the bind it. Okay, so bind it. So when the polymerase reaches some sort of phases, so DNA RNA hybrid result in the polymerase. So this is after that we can bind it, then normally the structure we can get it. Okay. Next we go for the process of transcription. See, this is the prokaryotic DNA dependent RNA polymerase, mRNA, tRNA. Provide template. Here coding template is there. Catalyze translation. Here RNA polymerase bind in the promoter region. Here transcription is the it is only initiators. In prokaryotes there are the 3 major types there: RNA, mRNA, transfer tRNA, messenger RNA. Then this is the recombinant RNA. That is the RNA. This is the full form of RNA. So what is that? mRNA means messenger RNA, tRNA means transcription RNA, and rRNA means recombinant RNA. Okay, all these RNAs are needed the synthesis of protein in cells. Very very important protein one. Here all is the catalytic role during the translocation. There is a single DNA dependent RNA polymerase that catalyzes the transcription of all types of RNA. It binds to the promoter and initiates the transcription. What is that? Bind, and it will be helpful for the transcription. Okay. So this is the polymerase binding sites are called promoters. Polymerase binding sites are called what? Promoters. Underline the word promoters. The uses of nucleoside triphosphate as substrate and polymerase in a template dependent fashion following the rule of the complementary. So the only the short stretch of the RNA remains bound to the enzyme. So polymerase reach that. See here, enzyme involved in the prokaryote transcript DNA dependent RNA polymerase. So ATP, GTP, adenine, thymine. This is the GTP, ATP. So protein will be added in the following of that. So prokaryotic transcription process here showing that. So in the prokaryotic processes, this is a sigma factor. Already yesterday class I told that is the what is a sigma factor and all. So in the sigma factor, the promoter is the polymerase enzyme. It's a DNA helix. So it is 3 is very important for the initiation. Then the elongation is after the termination. Termination means cut that. Okay, we can cut the termination part. So RHO factor is then now I told that before that and all. So RNA polymerase, it is helpful for the remainder. Okay, this is the formation of the prokaryote transcription process. So that question is how the polymerase are able to catalyze the 3 steps means these are the one is elongation and transcription and what is that? Here initiation, elongation, termination. That's all about it. First it will be started, after that it will be moved for process, and after that you cut it and take it out. Okay. So this is the bacteria since mRNA do not recur. What is that? Do not recur any processing or become active. So it will be separated for cytosol. This is because of genetic material is not separated from other cell organelles in the nuclear membrane. Eukaryotes there are at least 3 RNA polymerases. How many RNA? 3 polymerase there. So after that it will be helpful for the RNA material. So in eukaryote, the monocyte structure, genes have a coding sequence known as exons. It's very important. The coding sequence called exons, and the introns. See this is the structure. You can see that this coding is exon and intron middle, and after that termination. Okay, this three is very important for the messenger RNA DNA. Okay, so the introns are removed by the process called splicing. So introns we want to remove means splicing. Undergo the additional process called capping and trailing. Okay, so any process we want to remove, you can use the capping and trailing. In capping, an unusual nucleotide methyl granule and adenine residues 3 are trailing. Therefore, this process are now called as mRNA is transferred out of the nucleus for the translation. The split gene features of the eukaryotic genes is almost entirely absent in prokaryotics. Originally, each exon may have a code for the single and polypeptide chain with a specific function. Since exon arrangement and intron removal are flexible, so coding or polypeptide act as a domain common in various ways from new genes. Single genes can produce different functional proteins, and through the alternate splicing pattern, a mechanism known as play an important role in the generating the protein and functional diversity in animals. So how can we cut that? So already I told that spot is the meaning of splicing. So you can take it out from the exon means that is a splicing. So there is a exon will be cut the using of the splicing, and after that we are taking out from the exon part alone we will have separated. Here next intron and everything is the same, only the three pictures. So this is the arison before of the various protein in the evolution eukaryotic genes. If the introns arose later, how did they enter the eukaryotic genes? This question will be arise from that. So answer of this question is introns are mobile DNA. So it is a moving from one place to the target site. We can select it and we can eliminate the mediator, and HSGT lignase prokaryotic cells we can use that. So I hypothesize to have played a major role in the evolution of the life on earth. So thank you students. So this is the important question for that. So molecular genetics now over.